Ladies and gentlemen, welcome to this webinar on Spot-Tag. Spot-Tag is a small peptide tag for any kind of application, or in a nutshell, one for all. My name is Klaus Herrick. I'm the head of sales and marketing here at ChromoTek. I like to begin my presentation with a short outline of the content. I will start with introducing Spot-Tag and the corresponding Spot-Nanobody. I will also quickly explain what the nanobody is, and then focus in my presentation on applications. I will show examples on immunofluorescence and Super Resolution Microscopy. I will show examples on Western blotting, immunoprecipitation, as well as protein purification. Last but not least, I will provide an example on how to CRISPR spot into the genome of a cell and how to detect it both biochemically and with using a microscope. Finally, I will summarize those data and conclude my presentation. The quest for a new peptide tag. How does a perfect peptide tag look like? It should be short, it should be flexible and unstructured, it should be inert. You may want to fuse it to the N or both to the C terminal of your protein of interest. It should be highly specific, particular to the nanobody that detects it. It should have a low background, should not contaminate. It should work for any kind of protein. It should work for any kind of applications like immunofluorescence, immunoprecipitation, or protein purification. And of course, it should be cost effective. We do believe we have that protein tag and the corresponding Spot-Nanobody or VHH. How does the spot tag look like? The spot tag sequence is given here. It is a small size 12 amino acid peptide. It is 1.4 kilodalton in size. It has a PI value of 10.4. It is hydrophilic and very inert. We have characterized it. The molar extinction coefficient is 5,500. It is easy to clone. You get good expression yields. It works both on the N and C terminal side of your protein of interest. And so far, we and our customers have tested in bacteria, in yeast, mammalian cell lines, plant as well as insect cells. It is derived from beta-catenin and has been engineered for higher affinity binding. It is detected or bound to the highly specific Spot-Nanobody. What is a Spot-Nanobody or VHH? I would like to first introduce what a nanobody or VHH is. A nanobody is the binding domain of an alpaca heavy chain antibody. So alpaca possess normal IgG type antibodies as well as these heavy chain antibodies. And the binding domain of these heavy chain antibodies are called nanobodies or VHHs. They are just 15 kilodalton in size or about 2 to 3 nanometers. Now, when you conjugate a nanobody, or in this case a bivalent nanobody, to a fluorophore, you can use it for fluorescence and microscopy applications. When you conjugate it to a bead, you may use it for protein precipitation or protein purification. The Spot-Nanobody is fully characterized. It binds to the spot tag with a very high specificity and high affinity. The small size of just 14.7 kilodaltons, 2 to 3 nanometers, or in a bivalent format, just a little bit more than 30 kilodaltons or 4 to 6 nanometers in size. If you compare that to a classical IgG antibody, that is about 150, 160 kilodaltons. That is about 10 times larger than the nanobody. The Spot-Nanobody has a very high affinity to the spot tag of just 700 picomolar. There are strong avidity effects if the bivalent format is used. You may expect an affinity improvement by a factor of 400. The binding mechanism has been elucidated. I will show that on the next slide. And the nanobody is produced recombinantly without lot-to-lot variations. So what is the molecular mechanism of this high affinity binding? The Spot-Tag, as you can see here, is embedded on the surface of the Spot-Nanobody, which is this green surface that you can see here. The Spot-Tag becomes part of the beta sheet structure of the Spot-Nanobody. The specificity is achieved by multiple interactions of the Spot's nanobody side chains to the Spot-Tag, here in this cleft. And finally, the Spot-Tag is locked by two amino acid side chains. There is a glutamate and an arginine side chain that actually clamp the Spot-Tag to the Spot-Nanobody. As a result, you have a high affinity and high specificity binding of the Spot-Nanobody to Spot-Tag. The Spot-Nanobody has been rigorously characterized and is very stable. I will now provide some examples. For instance, the melting temperature determined by differential scanning fluorimetry is about 63 degrees. There is no aggregation observed for the Spot-Nanobody over a large temperature gradient from 20 to more than 80 degrees. The chemical stability is also outstanding. So here you can see the stability in increasing concentrations of urea, and you can see that the Spot-Nanobody is very stable even in a chromotropic reagent like urea. So in summary, you can say the melting temperature is about 63 degrees. It has an unusual high colloidal stability, and it is highly resistant to chromotrophs. Now I would like to move into applications of Spot-Tag. And I have to first introduce dedicated Spot-Nanobody tools for those applications. So here in yellow I have my protein of interest, which has been tagged with the Spot-Tag, the 12 amino acid sequence that you can see here. Now, for immunofluorescence, Super Resolution Microscopy, and Western blotting, you may want to use the Spot label, which is a bivalent format of the Spot-Nanobody that is labeled to a fluorophore. For immunoprecipitation, you may want to use a Spot-Nanobody that is conjugated to an agarose bead, to a magnetic agarose bead, or a dynabead. And finally, for protein purification, you may want to use a dedicated resin where the Spot-Nanobody is also conjugated to a magnetic agarose, however optimized for protein purification and gentle elution. Starting with immunofluorescence and super resolution microscopy. So we are looking at the protein of interest tagged with the Spot-Tag, and we are using a bivalent format of a Spot-Nanobody that is conjugated to a fluorophore, as we call it, a Spot label. The Spot label has high sensitivity and very low background. It provides high resolution because the size of the nanobody is rather small. You have a very small epitope label displacement, which is particularly important for super resolution microscopy. How does it look like? Here we have the Spot label of HeLa cells that are expressing an actin chromobody. So what you can see here is a control signal of EGFP. Here we have the Spot label labeling the actin, and here we have the merge of the EGFP signal and the Spot signal of the actin of HeLa cells counterstained with DAPI and merged. On the right hand side you can see confocal images of HeLa cells that are expressing Tom70 EGFP Spot-Tag. On the left hand side you can see the EGFP signal as a control, In the center you can see the spot label signal, and on the right you can see the merge. And you can see the merge is perfect. You have a perfect overlay of the spot label and the control EGFP signals. In super resolution, you get these great images like this. On the left-hand side, this is a HeLa cell's actin shown at a very high resolution using the spot label and the Spot-Nanobody, and on the right-hand side, you have the staining of vimentin spot in HeLa cells. It's just beautiful images playing with a card of high specificity, low background, and small size nanobodies. You may also use the spot label for fluorescent Western blotting. We do provide spot label conjugated to ATO488 and ATO594 for fluorescent detection in Western blot. Alternatively, you may use the spot nanobody plus a secondary anti-llama or anti-alpaca HRP conjugate. To our experience, the fluorescence detection is more sensitive. As you can, an example is given here for a GFP protein that has been tagged with spot and detected with ATO488 labeled spot label. Now I would like to introduce immunoprecipitation using the Spot-Tag and Spot-Trap, which is a spot nanobody conjugated to a bead. It provides high affinity binding. You will not have any antibody heavy and light chains that you may observe when you do an immunoprecipitation using conventional IgG style antibodies. You have a high specificity and a low background, and you can apply very harsh washing conditions if needed because of the extraordinary stability of the spot nanobody. How does it look like? First, here on that SDS gel, you can see the input lanes, the flow-through lanes, and the bound lane, and then confirmed by Western blot. So you can see the band of spot here in the input as well in the bound fraction. There is nothing left in the flow-through, and you observe a very nice, a very selective binding of spot, a very nice pull-down of spot. This shows a very effective immunoprecipitations are possible, and what's also very important is that we offer a variety of matrices, so the spot nanobody, the spot trap if you like, can be ordered as agarose, magnetic agarose, or dynabeads. I like to stress out the point that there is no heavy and light chain contaminations of an antibody that you would have like when you use a flag tag and do the pull down using a flag antibody, conventional antibody. The spot trap has and the spot tag have the best background in class compared to other affinity tags. So what have we done? We have done a benchmarking against other common affinity resins, and then we have done this immunoprecipitation from hex cells, lysates without any tags, and just looked into the background. So what you can see here is an SDS gel, and what has been done on this hex cell immunoprecipitation just with the resins, with the beads named here from various suppliers, and we've looked into the background. So here we have a glutathione cellulose from supplier 1, then we have an anti-MYC antibody, from a different supplier, we have FLAC resins used, we have dynabeads coupled to streptavidin used, and we've used spot trap, magnetic agarose, and spot trap agarose. And as you can see, every resin has a background, but the background of spot trap, magnetic agarose, and spot trap agarose is actually the lowest. So here you have hardly any background, And this is an indication on how powerful and specific the pull down of spot-tagged protein with spot trap is possible. You can also apply very harsh washing conditions, and these harsh washing conditions for the wash step in the immunoprecipitation maybe 2 molar sodium chloride, 2% nonidate P40. It is a bit sensitive on SDS. It can stand 0.1% SDS, but 1% Triton X100, 1% desoxycholate, 10 millimolar DTT, 0.75 molar guanidinium hydrochloride, 2 molar urea, and it is stable in a pH range from 4 to 10. You can see here these data as tested with M-sherry conjugated to spot. So here we have 1 molar, 2 molar sodium chloride, different series of detergents, and here we have the control, and you can see there is hardly any difference. Here we have tested various concentrations of urea, up to 2 molar urea. There is no difference to control, and same holds true for different concentrations of the reducing reagents DTT. Now I would like to introduce protein purification using the Spot-Tag. So again, we have our protein of interest that is tagged with a Spot-Tag. And here we have a dedicated nanobody, Spot-Nanobody conjugated to beads that we call Spot-Cap. Spot-Cap is in a manner modified that it is peptide elutable even at temperatures of 4 degrees in a very effective manner. So you get high purity in a one purification step. It is a gentle native peptide elution that results in high yields. You have almost no host cell contamination, and what's very important for being cost effective is you can apply multiple regeneration cycles. What you can see here is SDS page of the elution of a spot-tagged protein with spot peptide, and you can see here the input, that is kind of the wash or flow through, first wash. Here we have the elution applying 100 micromolar of spot peptide, and then we have done 5 elution steps, and you can see there is a high yield, 10 milligram of protein of a 30 kilodalton protein can be bound and purified by milliliter of settled resin. What's remarkable is that there is hardly any background. It's highly specific. These faint bands that you can see here are degradation bands of the spot cap. Now, the background has been also investigated using different host cell types. So we've looked into the background of mammalian, yeast, insect, and bacteria cells. So what you can see here is the incubation of different cell lysate expressing no-tag protein applied to the spot cap, and then you can see the lysate on the left, and here on the right you can see the eluted fractions, and you can see hardly any background. Here are 2 little bands on the yeast, a few faint bands here on mammalian, but on insect and bacteria there is no background of host cell proteins that may contaminate your purification of your spot-tagged protein. What's also important for protein purification is cost. These resins are expensive, and you may want to use them multiple times. In order to use them multiple times, they need to be effectively regenerated, and their binding capacity and binding specificity and their binding performance needs to be maintained during these generation cycles. So here we can see at the control R0, that's the elution without regeneration, and here is the elution fraction after 1, 2, 3, and 4 regeneration cycles with 100 millimolar glycine pH 2.0 each. And you can see there is no change in performance. So you get a high yield even after 4 regeneration cycles. And this is very cost effective because you can actually reuse your spot cap resin again and again. Now I like to show that CRISPR'ed Spot-Tag application. What has done in these experiments is the introduction of a Spot-Tag and an EGFP to the Lamin AC locus after start codon in HeLa cells. And we have conducted first an immunoprecipitation. You can see here the Western blot. Here we have the input, and here we have used here we have used a spot trap agarose, and as a comparison, GFP trap agarose. GFP trap agarose is a immunoprecipitation reagent that is also a nanobody against EGFP, and you can see there is no protein in the flow through, but there are 2 bound or immunoprecipitated bands for Lamin A and C in the bound fraction based on the interaction to the GFP trap, to the nanobody of the GFP trap against GFP, and here flow through and bound fractions of a spot trap fraction where you can see that the spot trap is equally performing as the GFP trap. Then we have conducted microscopic studies using immunofluorescence, and what you can see here is on the left hand side is the EGFP signal, and on the right hand side you can see the Spot-Tag signal, and you can see that these 2 images are perfectly colocalized at the nuclear lamina. So it's a very, very strong and powerful application to show how good spot-tagged proteins can be investigated. Now I'd like to summarize. The Spot-Tag and the Spot-Nanobody are a very versatile system for multiple capture and detection applications. You can use it for immunoprecipitation. It has a high affinity, it has a very superior low background, and you can apply stringent washing conditions when needed. You may use it for mass spec analysis, for instance in co-IPMS experiment. You may CRISPR-Cas the short tag relatively easy into the genome using CRISPR-Cas technology. You can use a Spot-Tag in Super Resolution Microscopy, where you obtain a higher resolution and a higher label density than with conventional antibodies due to the small probe size. You can effectively use it for protein purification with a spot cap. We have a high capacity, we have a native elution at 4 degrees and multiple regeneration cycles. You can use it for immunofluorescence, where the spot nanobody, the spot label, better penetrates the tissue and has a better access to the epitope. And finally, you may want to use the spot label for Western blotting to detect small nanogram quantities. Now I'd like to conclude my presentation and show those bodies that are very important for the development of nanobodies, which are the alpacas. I like to stress it is for research use only, and the other disclaimers here on the right hand side. I'd like to conclude my presentation by saying thank you for your attention.